Okay, today we'll study the next stage of the development, that is the third week of development. Okay, this is also called as the gastrulation. Okay, where the trophoblast will be modified. Okay, this is the third week of development, which is also called as the gastrulation. So these are the objectives which we'll cover. So we'll talk about the gastrulation. So what exactly you mean by gastrulation? Gastrulation is the process by which all the three germ layers are formed. Till now we are seeing that the embryo has two layers. It was one layer, it was embryoblast, then it changed itself into bilaminar disc, and the upper cells became the epiblast and the lower cells became hypoblast. Similarly, there were changes we saw within the trophoblast, which changed into cytotrophoblast and syncytotrophoblast, and the many modifications here. The blastocyst cavity became the primary yolk sac, then it became secondary yolk sac. Okay. Then similarly, there was appearance of the amniotic cavity. Now it is completely formed. Okay. Then it was connected by the connecting stalk. Now we are seeing the changes within the embryo. Okay, so till now we were studying that it was two layer. Okay, now this changes into trilaminar, that is three layer. So this process of changing from the two layered structure to the three layer structure, this is called as gastrulation is the process by which all the three germ layers are formed, as I was telling before. So the all the body structures, okay, are formed from three of these, any of these layers. There are only three layers: ectoderm, mesoderm, and endoderm. Okay, so all the body structures are formed from any of these three layers. Okay, so now all the three germ layers are formed within this embryo, which is still in the form of the a disc. If you see, this is how it looks like. So oval shaped structure. So this is called as the bilaminar disc. Now it is changing itself into three layer, that is called as trilaminar disc. Okay, so it changes from two layer to three layer. This is called as, this process is called as gastrulation. So now there are three layers: ectoderm, endoderm, and the mesoderm. So are well established in an embryo. So this is the embryo. Now it has changed from the two layer to the three layer. It is the most characteristic event of the third week of gestation. So this process of gastrulation is the most important feature of the third week of the development. So third week, three layers. Okay. It begins with the formation of the primitive streak. If you see here, if you this gastrulation, now we'll discuss how exactly the two layer structure has changed itself into the three layer. Now, if you see this embryo, two-layer bilaminar disc, if you see here, this is how it looks like. As I said, it is oval shape with the two layers. The upper layer is called epiblast and the lower layer is called the hypoblast, and it is oval shaped structure. Now, there is formation of something called as primitive streak. If you see here in the midline, there is appearance of a streak. This is called as the primitive streak on the surface of the epiblast. Epiblast means upper layer. Okay, epiblast, which will be clearly visible at the 15th or 16th day, though formed earlier. So even though it is formed earlier, it start appearing on the 15th or the 16th day. So by the 14th day, the second week has finished. Now by the 15th or 16th day, there is formation of the primitive streak. The cephalic end of the streak is called as the primitive node. So this is more formed at the caudal end, at the tail end. There is formation of a streak in the midline called as the primitive streak. Okay. The caudal, the cephalic means head end of this primitive streak, this midline structure will show a node that is called as the primitive node, which shows slight elevated area around a small primitive pit. In the center, there is a small pit, and around there is a thickening that is called as the primitive node. This is the primitive streak. This is a midline structure near the caudal end. Caudal means towards the tail end. So now with the formation, till now we did not know which is the head, which is the tail. It was like a germ disc like this with just two layers. Okay, but Now, with the formation of this primitive streak, we now know that this is the tail end. So, because the primitive end will formed at the tail end, the caudal end. Okay. So this primitive streak, if you see from the initially, will I will show you the pictures how it begins from two-layer to the three-layer structure. The embryonic disc will be oval in shape as we saw before, and later pear shape where the large end of the pear is at the cranial end and the small end is at the caudal end. Here also, if you see here, the larger pear shape, this is almost looking like a pear shape with the enlarged part. The bigger part is can be made known that this is the head end, and the thinner part, the taper end, this will be the tail end. Okay. Also, the primitive appearance of the primitive streak also shows that is the tail end. Okay, so embryonic disc will be oval in shape. Later, it becomes pear shape, where the large end of the pear is cranial end, and the small end is called as the caudal end. In the cranial part of the embryonic disc, the cells of the hypoblast become tall columnar, forming a thickening called as the procaudal plate. Very important. If you see here, this is it is shown round, but it is oval in shape. If you take a section at the midline like this, or if you see this picture, if you take a section at the midline, initially before the formation of the primitive streak, if you see, it is two layer, upper layer epiblast, which are columnar, and hypoblast cells will be cuboidal, as you can see in this picture. So epiblast above and hypoblast below. Okay, now if you see at the hypoblastic cells, slowly near the head end there is proliferation of these hypoblastic cells. Here they are all equal, but near the head end, there is proliferation of cells of the hypoblast, not the epiblast, hypoblast. In the cranial part of the embryonic disc, the cells of the hypoblast become tall columnar. Hypoblast cells become tall columnar, forming a thickening called as the procaudal plate. Now it is called as procaudal plate. If you see on the surface, it looks like this. Okay, so this is the midline section. So now this is called at the end. With the appearance of this procaudal plate, now we know this is the head end, this is the tail end, which is firmly attached to the underlying epiblastic disc, where the over membrane will be formed on the 14th day. So if you see here, these hypoblastic cells become tall columnar, and this is called as the procaudal plate. So if you see, this procaudal plate is totally adherent to the epiblastic cells, and this region will later form the oral membrane. Very important. This is the site for the formation of the oral cavity. Oral cavity. Okay. With the appearance of the procaudal plate, the pole of the embryo is determined. So before the primitive streak is formed, this appearance of this procaudal plate itself will determine which is the head end, because this procaudal plate is formed near the head end. Okay. And also the central axis, also the midline, which is the midline. Till now we did not know which is the midline. Now with the proform, because this is formed at the midline, so we know the midline, central axis, right and left half. So we know this part will form the right part of the body, this will form the right left part of the body. So now all these determined. This become the cranial end and the tail end. So this, where the procaudal plate is formed, this form the head end, and the other part becomes the tail end. Now it is determined. Okay. The cells of the ectoderm in the central axis near the tail end, caudal end, proliferates and elevates. This elevation is called as the primitive streak. This is the next stage. What we saw here, the primitive streak is the next step. First, there is formation of the procaudal plate, which is also called as the buccopharyngeal membrane. If you see later, it is called a buccopharyngeal membrane, and this is the site for the formation of the oral cavity. Now after this formation, now there is formation of the primitive streak in the tail end. Okay. So the cells of the ectoderm. Now till now there was enlargement of the hypoblastic cells which form the procaudal plate. Now there are changes taking place in the epiblast, which is also called as the ectoderm. Ectoderm. So if you see here, so now there is changes taking place within the ectoderm or the epiblastic cells. Cells of the epiblast or the ectoderm in the central axis near the tail end, caudal end. It is also called as caudal or the tail end, proliferates and elevates. If you see here, they start proliferating and getting elevated. This elevation is primitive streak. This is the primitive. So if you see on the surface, this is how it looks like. So this is the procaudal plate and this is the primitive streak. Procaudal plate formed near the head end and the primitive streak is formed near the tail end. Okay. It acts as the primary organizer and induces the formation of the intra-embryonic mesoderm and notoderm. So this primitive streak now will form something called as till now it was just two layers, epiblast as you can see here and the hypoblast. Okay, with the formation of the primitive streak, now the cells from the primitive streak start proliferating and passing between the ectoderm or the epiblast and the hypoblast. Now this is called as the mesoderm. So the cells of the cells of the ectoderm in the central axis near the tail end proliferates and elevates. This elevation is called as primitive streak. It acts as a primary organizer and induces the formation of the intra-embryonic mesoderm. So now the intra-embryonic mesoderm is being formed and the notochord. So disappears by the 26th day of development. Afterwards, once this mesoderm is formed, this primitive streak starts disappearing by 26th day. Okay, everything whenever we mention about the day, it starts from the 0 day. 0 day means fertilization. From that day up to the 26th day. So by the 26th day, the primitive streak starts disappearing. The proliferated cells start migrating sidewards and pushing epiblast and hypoblast. As I said, from the primitive streak, the cell starts proliferating, moving outwards. Okay, so they start moving outwards and they pass between the epiblast and the hypoblast. So epiblast is called ectoderm and hypoblast is called as now endoderm. And this cells proliferating from the primitive streak, these are called as the mesoderm. So now there is formation of three layers. So it started from the two-layered structure. Now it is becoming three-layer. This process of formation of three layers is called as gastrulation. Proliferated cell starts migrating sideways, pushing the epiblast and the hypoblast. As I said, here the epiblast and the hypoblast cells move outward. Till now they were totally adherent. Now because of the mesoderm, so they start moving outward and in between there is formation of one more layer called as the mesoderm. This inward movement is known as invagination. So they start invaginating in between the ectoderm and endoderm. So this process is called as invagination. So now if you see here, is the same oval shaped embryonic disc which was two-layer with the formation of the primitive streak. Now the cell starts proliferating. If you see here, the same two-layered structure. This is the epiblastic cells and these are the hypoblastic cells. Hypoblast and these are the epiblast. Now from the midline, that is the primitive streak near the tail end, now the starts proliferating. If they are moving sidewards, and now this is the mesoderm. So the epiblast now will be called as the ectoderm and the hypoblast will be called as the endoderm, and this cells from the primitive streak, now these form the mesoderm. So now it is the three-layered structure. Cell starts migration and specification is done by the fibroblast growth factor 8. So this is a very important factor which will help in the determination of the cells. Okay, still they are just cells. We don't know. Still there is no organ, no formation of head, brain, nothing is there. No limbs, no abdominal organs, nothing. So it is just the cells, but they start differentiating by the there is a growth factor 8 fibroblast which specify each cell with a specific function, which is synthesized by the primitive streak itself. So here is the primitive streak and here is the primitive node. Okay, and the primitive pit inside. Okay, this is the primitive node and within that there is primitive pit, this depression. After invagination, some cells displace hypoblast and create embryonic endoderm, and others lies between the epiblast to form the intra-embryonic mesoderm. Okay, so there is differentiation of the endoderm and the mesoderm. The cells of the epiblast then form the ectoderm. So these epiblastic cells now form the ectoderm, hypoblast become the endoderm, and these cells form the mesoderm. So all the three germ layers are formed from the epiblastic cells. So if you see actually, because the primitive streak has formed from the epiblast, okay, so the mesoderm is formed from the epiblast, The epiblast itself is also formed from the epiblast. Even if you see the hypoblast, so that are also initially these two were one. So even the hypoblasts are formed from the epiblast. So all the three germ layers are actually formed from the epiblastic cells. So epiblastic cells are also called as the stem cells. Stem cells are those cells from where all other cells are determined. Okay. So the most primitive types of cells are called as the stem cells. This process of formation of the primitive streak, endoderm and intraembryonic mesoderm is referred to as gastrulation. As I said, so this formation of three-layered structure of the embryo, this is called as the gastrulation. The intraembryonic mesoderm spreads everywhere in the disc except in the procaudal plate where the ectoderm and endoderm are adherent without the mesoderm in between. If you see here, as I said, so this mesoderm will be spreading everywhere, as I said. It will spread everywhere except the procaudal plate. Here, this is the procaudal where the oral cavity is formed. So there it cannot enter because these cells here, they are totally adherent. Okay, so let me show you. Okay. So if you see here, the epiblast and the hypoblast are totally adherent in the oral cavity, this procaudal plane. So there it cannot enter. Every other place the mesoderm is formed, but not in the procaudal plate, which is also called as the oropharyngeal membrane or buccopharyngeal membrane. Many names are there. Okay. So these cells cannot migrate into the procaudal plate. Everywhere else they will go. Okay, mesoderm is formed everywhere except the procaudal plate where they are adherent. So that's why it remains thin because the epiblast and hypoblast are totally adherent. So it will remain very thin. This will later form the buccopharyngeal membrane. As I said, this uh, procaudal plate will form the buccopharyngeal membrane or oropharyngeal membrane or oral cavity, whatever you call it as. Okay. Similarly, areas exist caudal to the primitive streak at the site where the ectoderm and endoderm remain contact. So similarly, even behind the primitive streak, there is an area where the cells will be totally adherent, ectoderm and endoderm. So there is no mesoderm. So this will form the cloacal membrane. This will form the cloacal membrane. So now there are two membranes here, procaudal plate where there is buccopharyngeal membrane formed. Here this is the cloacal membrane. So what is the importance of cloacal membrane? It will form the future urogenital region. So this will form the urogenital region. This will form the oral cavity mouth. Okay. The connecting structs become comparatively smaller with the growth of the embryo and comes toward the tail end. If you see here still if you see here, this is the amniotic cavity, this is the secondary yolk sac. It was two-layer epiblast and hypoblast. Now with the formation of primitive streak, there is third layer called as the mesoderm. And here there is the connecting stalk which comes toward the tail end. Now there is formation of a very important structure which is called as the notochord. Notochord. This is formed on the 17th or the 18th day. Okay. Notochord is a midline structure develops from the upper end of the primitive streak to the lower end of the procaudal plate. If you see here, so here from the upper end of the primitive streak and to the lower end of the procaudal plate, there is formation of something called as the notochord. Notochord. So in the midline, okay. So notochord is a midline structure developing from the upper end of the primitive streak to the lower end of the procaudal plate. This is formed on 17th or the 18th day. Now primitive node or primitive knot of Hansen knot, which is the upper thickened end of the primitive streak. If you see here, so this is called. So let me show this picture. Okay, here if you see here, this is called as the primitive node. This is the primitive streak. The upper end of this primitive streak is called as primitive node or primitive notch, or even it is called as primitive pit. All the same. Okay, so this is the upper end of the primitive streak. The primitive pit depression in the center of the primitive node. So there is depression within the primitive node. This is called as the primitive pit. Then notochordal process. Cells multiply in the midline between the ectoderm and the endoderm, reaching the lower end of the procaudal plate on the 17th and the 18th day. This is called as the procaudal process, as we saw before. So Here, this is called as the procaudal process. Then the notocaudal process, sorry. The notocaudal canal, within this notocaudal process, the primitive pit extend into the notocaudal process, converting it into a canal. If you see here now, to better appreciate, so there is the epiblastic cells, these are the hypoblastic cells. At the procaudal plate, the epiblast and the hypoblast are totally adherent. This is the mesoderm now. This is the ectoderm, endoderm, and this is the mesoderm. Here is the primitive streak. At the upper end of the primitive streak, this is called as the primitive knot. Now there is a depression called as the blastosphere, or it is also called as the primitive pit. Now from the primitive pit, there is formation of something called as the notocaudal process in the midline. Okay. So now this primitive pit will extend through this notocaudal process. Now this is called a notocaudal canal. So the same thing if you see from above. So here this is the procaudal plate, this is the cloacal membrane, this is the head end and tail end, this is the primitive streak. At the upper end of the primitive streak, you can see this is the primitive knot. So now there is formation of a depression called as the blastosphere or the primitive pit. Now there is an extension from the primitive pit in the midline up to the oral cavity. This is called as the notocaudal process. Initially it will be notocaudal process. With the canalization of this is called as the notocaudal canal. So this is all about this. Okay. The floor of the notocaudal canal breaks down and then the notocaudal canal communicates with the yolk sac and also amniotic cavity through the primitive pit. Now what happens? Now it will break down in the lower part. Upper part is already open. The lower part here it all breaks down. Now if you see here in the picture, so this is the same embryo. These are the epiblast, and here they are the which form the ectoderm. Here is the endoderm, and in between is the mesoderm. So here there was notocaudal process formed, then the notocaudal canal. Now the floor of this disappear. Now this notocaudal canal starts communicating with the, this is the secondary yolk sac. If you remember, this is the secondary yolk sac, and above is the amniotic cavity. If you remember this picture before, so above here is the amniotic cavity, below is the secondary yolk sac. So there is communication now between the secondary yolk sac and the amniotic cavity. Okay, so now there is communication between the amniotic cavity and the secondary yolk sac because this floor has disappeared. Okay, so through this primitive pit, now there is communication. Later, again it closes. So initially it will be in the form here, there is formation of the notocaudal plate. Now later it closes. This will close and it form the notochord. So the notocaudal plate, if you see here, this is the notocaudal plate. Cells of the notocaudal canal becomes flattened. So the cells here become flattened and it forms a plate. Then there is formation of definitive notochord. This is the definitive notochord. Proliferation of cells forms solid rod of cells. Now it closes, this communication closes. Now it becomes this notochordal process plate becomes a solid cord-like structure. This is called as the notochord. Okay. So why this notochord is important? We're studying in so much detail. So it is in the midline. It is a midline structure between the ectoderm and endoderm in the same level as that of the mesoderm. Okay. So this is the mesoderm here, and this notochord mesoderm is everywhere except the midline. Midline we have the notochord. What is the importance of this notochord? Notochord acts as the forerunner in the development of the vertebral column and induces the formation of the neural tube, a very important structure called a neural tube from where the brain is developed. So notochord acts as a forerunner. It helps in the formation of the development of the vertebral column, the backbone. So these are the vertebral column, and the induces the formation of the neural tube. Later we'll see the neural tube, how it is formed and how the brain will develop from this neural tube. So the notochordal cells persist in the first decade of life as nucleus pulposus of the intervertebral disc. So this notochord will induce in the development of the formation of the vertebral column and the neural tube, which will form the brain. Later it disappear. Except it remains as a small structure within the vertebral column, which is called as the nucleus pulposus. If you see, this is the intervertebral disc. Between the vertebrae, there are spaces which are filled with a structure called as intervertebral disc. Intervertebral disc is made up of the intercentral part called as nucleus pulposus, and outside we have the annulus fibrosus. So the notochord will be remain as a remnant. So it will totally disappear except a small remnant which will form the nucleus pulposus in the first decade of life. Later it calcify and it will form much more stronger structure. Now, formation of neural tube. I said the notochord is the forerunner in the development of the neural tube, which will later form the brain. So now we are seeing the development of this neural tube. If you see here, this is the neural tube. Formation of the neural tube. So if you see here, so this is the notochord red line, and above that is the formation of the neural tube, neural plate. Initially, it will be neural plate, then it forms the neural tube. Okay, then it helps in the formation of the brain. Formation of the neural tube. So neural tube is derived from the ectoderm, as you can see here. This is the upper layers. These are called as the epiblast, which will now form the ectoderm. These are the hypoblast, which will form the endoderm, and in between we have the mesoderm on sides, and in the midline there is the notochord. Okay, so now here we are seeing the changes in this blue epiblastic cells or the ectoderm. So neural tube is derived from the ectoderm neural plate. So initially it will be formed on the neural plate as we saw before. So here is the neural plate, notochordal plate. Above that will be the neural plate. Okay. Overlying the notochord, overlying above the notochord, as we said, above the notochord will be the neural plate, and hence extend from the procaudal plate to the primitive node. So just like the notochord is from the primitive strip up to the procaudal plate, similarly even the neural plate extends from the primitive streak up to the procaudal plate. The neural tube gives rise to cranial enlargement forming the brain, and the caudal tube forms the spinal cord. So if you see, this neural plate will enlarge in near the procaudal plate to form the brain. Okay, and the caudal part remains thin and it helps the formation of the spinal cord. Very important structures are being formed at the third week of development. So the development of the brain as well as the spinal cord. Okay, the process of the formation of the neural tube is called as neurulation. So this neural tube formation, this is called as neurulation, which will be on the dorsal aspect. Dorsal aspect means on the back. As we know, the brain and the spinal cord will be developing from the back, which is from the epiblast cells. Now, differentiation of the intra-embryonic mesoderm. Mesoderms are divided into two types. One is the extra-embryonic and the intra-embryonic. If you remember, so there was formation of the extra-embryonic mesoderm. We were studying the uh, mesoderm before. That was the extra embryonic mesoderm. If we remember, in the second week of development, whatever mesoderm we were talking about, that was the extra embryonic mesoderm. Within the embryo, now there is formation of this mesoderm, which we already saw. So this was the mesoderm. So this is the mesoderm developing from the primitive stage. This is the mesoderm which was spreading everywhere. This is the intra-embryonic mesoderm. Okay. So now this intra-embryonic mesoderm differentiate itself. Okay. So here, if you see here, this is the notochord, and beside that is the mesoderm. Mesoderm here, mesoderm, as well as here mesoderm. So now the mesoderm is differentiated itself into paraxial mesoderm. Paraxial mesoderm becomes segmented to form somites which lie on either side of the neural tube. So if you see on either side of the neural tube, the mesoderm which were there. So if you again go back to these pictures, if you see here, this is the notochord and here was the neural tube. And here this is the intra-embryonic mesoderm. Now this intra-embryonic mesoderm which is formed, it will start differentiating into three parts. This will differentiate into three parts. The part which is just beside the notochord, this will be called as the paraxial mesoderm. Axis is the center. Para means beside. Beside the axis, this is called as the paraxial mesoderm. So this paraxial mesoderm will later change itself into somites. Somites. Okay. Somites are triangular structures. If you see here from the, if you take a section at this level, this is how it looks like. Initially, they will be rounded here. So later, they form somites. These are the somites, which are triangular structures and has a cavity inside in between. Okay. Somites are 44 pairs, 4 occipital, 8 cervical, 12 thoracic, 5 lumbar, 5 sacral, and 8 to 10 coccygeal. So these somites are divided into three parts again. So the paraxial mesoderm itself is divided into three parts. Now within this paraxial mesoderm, the somites itself divide again into three parts. The first one is called as the sclerotome. They migrate around the neural tube and form the vertebral column and ribs. So this one part which is around the notochord, so and the neural plate here above, so they will go around the neural tube and form the vertebral column and ribs. This is called a sclerotome. Sclero means the bony. Okay. Then the second layer form the dermatome. As the name derma means, it will be outside the skin. So they line the deeper surface of the ectoderm, form the dermis of the skin. So they will be forming derma. So that's why they form dermis of the skin. Skin is made up of the epidermis and the dermis. Okay. So dermis is formed from the dermatome. And myotome, myo means muscle. They form the striated muscles, all the muscles of the back and other parts. So sclerotome, these somites are very important structure which differentiate itself into three structures. Sclerotome which form the bone, dermatome which form the lower part of the skin, and the myotome which form the muscles. So this is a very important structure called as the sclerotome, which is derived from the paraxial mesoderm. Paraxial mesoderm. This is how it looks like before, and if you take a section at this level, now with the development of somite, this is how it becomes triangular. Initially it will be rounded, now it becomes the triangular in shape. Now, beside that, there is the next part of the mesoderm. This is called as intermediate mesoderm, and then the third part, that is the lateral part, lateral plate mesoderm. So the mesoderm, as I said, it is divided into three parts: paraxial mesoderm, green structure; red structure is called intermediate, and the lateral plate mesoderm. So the second part is the lateral plate mesoderm. So now we are talking about this part, which is called as the lateral plate mesoderm. We have already talked about the paraxial here. Now we are talking about the lateral plate mesoderm. The lateral plate mesoderm give rise to small cavities appear which coalesce to form the intraembryonic coelom. Okay, so if you see here, there is formation of a cavity within this paraxial, the lateral plate mesoderm, within the lateral plate mesoderm. So initially it will be solid. Now there is formation of a cavity within that. So there is formation of a cavity. So if you take a section at this level, so you can see, okay, here is the somites, and this is the intermediate mesoderm. This is the lateral plate mesoderm. Within that there is a formation of a cavity. Okay. It is horseshoe shaped with the two ends joined cranial to the procaudal plate. So if you see here, it looks like a horseshoe with the upper end, cranial end is joined together above the formation of the procaudal plate and even the brain formation. This is the neural tube formation. Okay, above that is the communication between one side to the other side. So it looks like a horseshoe, communicates with the extra embryonic mesoderm laterally. Now slowly it starts opening and it starts communicating with the lateral plate mesoderm. If you see here, this is the embryo, the three layers. So it was closed here. This is the which is the intraembryonic coelom. Now it opens and it starts communicating with this is the extra embryonic mesoderm, which we were studying before. Extra embryonic. Now the intraembryonic mesoderm starts communicating with the extra embryonic mesoderm. Now it splits itself into three layers called as this planktopleuric, somatopleuric, and intraembryonic coelom. So the splanchnic layer, the upper layer of cells here. If you see here, this layer of cell. Okay, one is called from the somatofluoric, and the lower layer is from the splanchnic layer. Here, if you see here, this is the same thing, somatofluoric, and here is this splanchnic layer. So the splanchnic layer will form this splanchnic layer will form the cardiogenic area, which will later develop into the heart. The somatofluoric above this layer will form the ectoderm, and the intraembryonic coelom, this is the intraembryonic coelom, form the pericardial cavity, pleural cavity, and the peritoneal cavity. These are the three important cavities which we have in the thorax and the abdomen. Around the lungs, there is the pericardial, the pleural cavity. Around the heart, there is the pericardial cavity, and within the abdomen, we have the peritoneal cavities. All these cavities are formed from the intraembryonic coelom. Very important. Okay. Then there is formation of something called as septum transversum. If you see here, this is the future septum transversum. This is the unsplit mesoderm cranial to the cardiogenic area, which later forms the diaphragm and liver. Very important again, very important. All important structures are being formed now. Now there is slowly there is differentiation of the cells. Still they are not formed, but they will form in the future. So now they are determining which structure will form what. So this septum transversum, so here is the septum transversum, will later form the diaphragm as well as the liver. Okay, so this is till here. Now we will talk about the folding of the embryo and how the yolk sac become small and small in case of human being, and it totally disappears. As we knew, so above there is, this is the two-layered embryo which become three-layer now. Above is the amniotic cavity, below is the yolk sac, secondary yolk sac. Now see here, this small cavity, amniotic cavity, was initially here, like this yolk sac was bigger compared to the amniotic cavity. Now the amniotic cavity become larger and larger, and the yolk sac becomes smaller and smaller. So with the becoming amniotic cavity become larger, and the yolk sac becoming smaller and smaller, it starts bending, folding. So this is called as the folding of the embryo. Okay. So the folding of the embryo with progressive increase in the size of the embryonic disc. The embryo itself, this is the embryonic disc, still in the form of a disc. Okay. So as the embryo grows in the disc formation, it bulges upwards into the amniotic cavity. It bulges, if you see here, it is bulging into the amniotic cavity. Further enlargement leads to folding of the embryo. If the embryo now starts becoming larger and larger, it starts folding because there is no space. No, it starts folding itself at the head end and the tail end. It folds everywhere, but it is more at the head end and the tail end. These are the head and tail folds. Similarly, the folds are formed on each side to form the lateral fold. Similarly, there is also folding on the lateral side. So there is folding at the head end and the tail end. Now we know which is the head end. Okay, and the tail end. Now there is folding at the head and tail end as well as on the lateral side. Formation of the head and tail folds leads to enclosure of the parts of the yolk sac within the embryo and lined with the endoderm. So, with the bending, we'll discuss this again in detail later. So, this the endoderm, which was here, endoderm, ectoderm, endoderm, and mesoderm was here. Now, the endoderm will get encapsulated within inside. Okay, enclose of the parts within the embryo and lined by the endoderm. So this now cavity is formed, and this will form later the gastrointestinal system, the gut. This formed the primitive gut, which initially has wide communication with the yolk sac. Initially, this gut is formed and there is wide communication with the yolk sac. Later, it becomes small and small, and we'll discuss all these things again in detail in the future classes. This is the embryo straight. This is the bilamina, then it becomes trilaminar. This is the amniotic cavity yolk sac with the development of the embryo. So the this starts bulging, amniotic cavity starts surrounding, and the yolk sac becomes small and small. As the embryo folds itself in the head and tail end, this cavity here, this forms the gut, and there is communication with the yolk sac, but it becomes smaller and smaller. Now with the complete folding, it becomes very small, communication. So this secondary yolk sac now it itself form the definite yolk sac. So all these details again we'll discuss in detail. So I will not go into details about this. So this is again the embryo folding. We'll have a separate lecture on the foldings of the embryo where we'll discuss all this in details. Okay. So this is all about the changes which are taking place within the embryo in the third week of development. So the main change in the third week of development is the formation of gastrulation and the differentiation of the three layers, ectoderm, endoderm, and mesoderm, and the differentiation of the mesoderm into the uh, the the paraxial mesoderm, then the intermediate mesoderm, as well as the lateral plate mesoderm. Okay, so this is all about this lecture. If you have any doubts regarding this, you can ask me. Any doubts? So it is lot of information. You have to go back home again, read in detail all these three lectures, very important lectures, because as I said, these are all connected to each other. Next, again, it will next lecture will be more higher. Okay, so go back and read again all these three lectures. Repeat this first week of development, second week of development, and third week of development. Then you will understand the next lectures. Okay. So if you have any doubts regarding any part of these three lectures, you can ask me.
No,
Doctor,
doctor, thank you very much.
I want to ask you something, please.
Yes.
I really want to ask you how to study.
How to
Because
study?
it's so complicated and lots of words, and I
Yeah,
don't know
yeah,
from
yeah.
where should we start.
Okay. Start from the first week. Try to understand the first week of development. Okay.
Okay,
Once you understand that, go to the second week of development. You have to read it. If you once you don't understand, second time you read, third time you read. So you have to read again and again. And whatever you understand, if you can help your friend, try to explain to him or her, and they will in return they can explain you. So if you keep discussing, anatomy is mainly discussion. When you discuss and explain to others, you will understand better. Okay, this is how you can read and try to help each other so that you can all learn together. Okay, start with the first lecture, first week of development. Once you understand, then go to the second week. Once you understand that, then go to the third week. Okay, so they are, because they are all interrelated, and I know that is a little complicated because a lot of new information is coming up here. So it you might feel difficult initially, but once you repeat, read, and repeat it again, inshallah, it becomes easy for you. And
okay,
anytime if
thank you
you
so
don't
much.
understand any particular concept, I can explain you.
Okay, thank you, doctor.
Welcome. Anything else? Okay, then thank you very much.